Good day everyone! We are Group 5 and in this video, we present to you the experiment entitled Isolation of DNA from cheek cells. Before we commence with the actual experiment, let us first know a little more about DNA. Almost every cell in the human body contains genetic material. Within the nucleus is the deoxyribonucleic acid, or DNA, which contains our unique genetic code, the information needed to program our cells' activities. DNA is under the important class of biomolecules called nucleic acids. Structurally, DNA is a polymer made of smaller repeating units called nucleotides. Each nucleotide is made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. DNA, as its name implies, is a nucleic acid based on the sugar deoxyribose. The other type of nucleic acid present in organisms is ribonucleic acid, or RNA, which is ribose instead. The difference between the two sugars is that ribose has an extra hydroxyl group at carbon 2, while deoxyribose does not. In living organisms, DNA exists as a double-stranded molecule intertwined in a double helix shape, with its negatively charged backbone composed of sugar and phosphate groups, while the base is in one strand pair with a complementary base on the opposite strand through hydrogen bonding. In terms of length, the length of DNA compared to the cell that contains it may be 1,000 times longer. For example, the DNA in human cells may be stretched out for 2 to 3 meters in length, and DNA mostly exists in cells in a loosely packed form. However, one exception is prior to cell division. At this phase, DNA supercoils into chromosomes. This packaging of genetic material into cells via supercoiling is an important feat in order for DNA to be managed easier during cell division, which involves the segregation of DNA into daughter cells. This ensures that the complete genome will be present in the daughter cells. Moreover, DNA supercoiling inhibits DNA nodding, which interferes with the normal functioning of the DNA and may be detrimental to biological functionality. DNA isolation is the process of extracting DNA from biological samples. This is an essential first step in the study of DNA sequences, analysis of genome structure, and gene expression. Thus, the quantity, quality, and integrity of the DNA isolate is important. In eukaryotic cells, 90% of DNA is localized in the nucleus, while the remaining DNA is seen in other organelles like the mitochondria. Since DNA is only about 1% of the total mass of a cell, the purpose of DNA isolation is to separate DNA from other cellular components, most notably RNA and proteins. DNA isolation is of primary importance in the study of genetic causes of diseases, development of drugs in medicine, genetic engineering, and forensic science.
Here are the aims of the experiment. First and foremost, we must be able to isolate DNA from cheek cells. Then, we must determine the mode of action of the reagents stated in the procedure. And speaking of procedures, we are to compare the procedure executed in the experiment with standard lab protocols for DNA isolation. Also, we are to describe possible qualitative tests that will ascertain the presence of DNA in our sample. And of course, the latent objective of the experiment is to test our skills and resourcefulness needed in the practice of biochemistry.
Now let's move on to the methodology. To start with the at-home isolation of DNA from cheek cells, the following common household items were prepared: a clear cup or small container, 70% isopropyl or ethyl alcohol that has been stored in the freezer overnight, concentrated dishwashing liquid, 50 ml of drinking water, and table salt. The first step was to create a saline solution in the clear container by dissolving 1 tablespoon of table salt into 50 ml of drinking water. In preparation for gargling the saline solution, we cleaned our mouths and scrape our teeth against our cheeks for around 30 seconds. We then gargled the saline solution for 30 seconds and placed it back in the cup. Next, we added 3 drops of the concentrated dishwashing liquid and stirred it for 1 minute, carefully enough to avoid creating bubbles. After stirring, we poured around 50 ml of the ice-cold alcohol carefully along the sides of the cup so that it forms a layer on top. After letting it stand for 3 minutes, we then observed for the filaments of DNA and took photos of the DNA isolates. Our group isolated DNA from cheek cells, but if you want to isolate DNA from a banana, here are the materials that are needed. A clear cup or a small container, 50 ml of drinking water, 70% isopropyl or ethyl alcohol that's been stored in the freezer overnight so that it's ice cold, concentrated dishwashing liquid, coffee filter, table salt, and a Ziploc bag. The first step is to mash around 5 cm of overripe banana inside a Ziploc bag for 1 minute. And then, to the mashed banana, add 10 ml of salt solution or just enough to solubilize all the mashed banana. Next, add 3 drops of the concentrated dishwashing liquid and stir it for 1 minute. After stirring, filter the resulting solution using a coffee filter, and in a separate container, add 5 ml of ice-cold alcohol to 5 ml of filtrate through the side of the container. Finally, let it stand for 3 minutes, then observe and collect the filaments of DNA. The following materials and equipment are needed for the DNA isolation from bovine spleen: scalpel and blender, ice-cold pH 7.4 sodium citrate saline buffer, centrifuge, conical tubes, and water bath, 2.0 molar sodium chloride, SDS solution, beaker and glass rod, absolute ethanol, 70% ethanol, and finally, distilled water. The first step is to weigh and dice 25 grams of fresh and clean bovine spleen using a scalpel. Next, homogenize the tissue with 5 ml of ice-cold pH 7.4 citrate saline buffer per gram of spleen in a blender for 10 minutes. Next, collect the homogenized tissue and centrifuge it at 6,000 rpm for 10 minutes, after which discard the supernatant. Suspend the pellet in 10 ml of 2.0 molar sodium chloride in a conical tube and stir vigorously for 1 minute. Next, add 250 microliter of 0.2 gram per ml SDS solution and incubate in a 40 degrees Celsius water bath for 10 minutes. The next step is to centrifuge it at 6,000 rpm for 10 minutes, after which discard the pellet and transfer the supernatant in a beaker. Carefully pour an equal amount of cold absolute ethanol along the side of the beaker. And then, insert a glass rod until the bottom of the beaker and stir in small circles in a clockwise direction so that you could collect the DNA fibers. In another beaker, wash the collected DNA fibers with 70% ethanol. And finally, resuspend the DNA fibers in 10 ml of distilled water, label properly, and store at 4 degrees Celsius.
Sodium chloride serves the same role in both the household DNA isolation and DNA isolation from bovine spleen. It creates a hypertonic environment which aids in breaking the cells to release cellular components, including the DNA. Sodium cations from sodium chloride, as well as from sodium citrate and sodium dodecyl sulfate or SDS, form ionic bonds with a negatively charged DNA backbone. Since the phosphates of each nucleotide in the DNA contribute a negative charge, this makes the DNA anionic in physiological pH. Sodium cations neutralize and stabilize this negative charge. As a result, DNA easily dissociates from water. Thus, the introduction of sodium cations promotes the precipitation of DNA in the next steps. In addition, Sodium chloride helps purify the DNA by dissociating some non-histone and histone proteins from the chromatin. Lastly, sodium cations introduce steric hindrance when DNases try to attack the DNA molecules, thus protecting DNA from DNase degradation activity. Similar to other buffers, sodium citrate buffer resists pH changes. The pH 7.4 buffer keeps the solution at or near physiological pH. This maintains the structure and net charge of the DNA, which is utilized to precipitate it out in the later steps. Additionally, it maintains osmotic pressure within the cell to avoid premature cell lysis during homogenization. As mentioned, in the DNA isolation from cheek cells, concentrated dishwashing liquid was used to solubilize the lipid bilayer of the cell and nuclear membranes to release the genetic material. Likewise, SDS is a detergent that is more potent in breaking the membranes. It is also a denaturant as it can break the secondary and tertiary structures of proteins, which helps remove protein associated with DNA. However, it does not affect the DNA because of the electric repulsion between the two negatively charged molecules. Alcohols such as ethanol and isopropyl alcohol are the key to the precipitation of DNA. The sodium cations by themselves cannot precipitate DNA since water prevents them from completely interacting with the DNA phosphate backbone. In other words, water does not permit the electrostatic interactions between the charged molecules as the water forms hydration shells around these molecules. The addition of ethanol disrupts the hydration shell by forming hydrogen bonds with water, pulling them away, and thereby allowing sodium cations to bond with the phosphates. Unlike water, ethanol and isopropyl alcohol have lower dielectric constants. Dielectric constant is a measure of the ability of a substance to insulate charges from each other. Lower value translates to a better interaction of charges when the substance is in between. This explains why ethanol and isopropyl alcohol can better permit the charged DNA and sodium cations to interact with one another. As mentioned, the anionic DNA is stabilized by the cationic sodium. This makes DNA less hydrophilic and aggregate with one another, resulting in DNA precipitation. Upon a slow addition of alcohol to the sample solution with detergent, a layered mixture was formed as seen in Figure 2 because the alcohol top layer has a slightly lower density than the aqueous solution. The alcohol layer contains the DNA components, and it is important to prevent the mixture of the alcohol layer and the bottom aqueous layer. This is because, if alcohol were to mix with the solution, ethanol or isopropanol molecules will be scattered about and will themselves be surrounded by hydration shells, losing their purpose. When the interaction of water molecules is minimized, a translucent, white, fibrous, loosely condensed precipitate is formed. As seen in Figure 3, only a small yield of DNA precipitates is obtained, maybe because of 1. The accumulation of saliva, which decreases the attrition of mucosa and germline cells, which lessens the desquamation of cheek cells, and 2. The varying innate desquamation of cheek cells of each individual. Not only DNA may be precipitated when cold alcohol is added. Due to the inherently lower solubility of sodium chloride and isopropanol, salt precipitates are formed in the trials wherein cold isopropanol was used. On the other hand, when cold ethanol was used, negligible to no salt precipitates are formed, since sodium chloride has a relatively higher solubility in ethanol, which is 0.65 gram per kilogram of the alcohol, than in isopropanol, which is 0.03 grams per kilogram of the alcohol. In addition, the cold temperature of the isopropyl alcohol decreased further the solubility of sodium chloride. With this, Room temperature isopropyl alcohol is advisable for DNA isolation experiments which deal with samples with low DNA concentration which must occur fast at room temperature. On the other hand, cold ethanol is advisable for DNA isolation experiments wherein the samples are assumed to have high DNA concentration and the DNA isolates are to be stored in a cold environment for a long period of time, such as in the isolation of DNA from bovine spleen. The isolated DNA is washed with 70% ethanol to remove the remaining salt residues and other water-soluble impurities, which can interrupt with the resuspension of DNA in water for storage. However, DNA is commonly resuspended in buffers such as TE buffer, which contains tris and chelating agent EDTA to inhibit the degradation of DNA by DNases.
Typically, the final step of any DNA isolation procedure involves the evaluation of the concentration and purity of the DNA collected, which is done using ultraviolet spectrophotometry. The pyrimidine and purine bases of DNA are responsible for the absorbance of the light and shows DNA concentration which is measured at 260 nanometers, as this is the maximum absorbance of the bases. The linear relationship of DNA absorbance at 260 nanometers and DNA concentration N is given by the equation N is equal to the absorbance at 260 nanometers over epsilon, where epsilon is the DNA extinction coefficient. On the other hand, DNA purity is determined by measuring absorbance at wavelengths other than 260 nanometers. For example, the absorbance at 320 nanometers should give the degree of a contamination of a DNA sample, since DNA does not absorb light at this wavelength due to light scattering. However, a UV spectrophotometer was not used in this experiment. Thus, another way of determining purity relatively is by comparing the methods and reagents used in the different protocols. In comparing the DNA isolation methods for cheek cells and bovine spleen mentioned earlier, it is expected that the reagents and equipment used for the latter may result in a DNA isolate being relatively purer. Firstly, the bovine procedure made use of additional reagents such as the citrate saline buffer, which ensures the stability of the pH throughout the experiment. Second, in terms of the solubilizing agents used for cell lysis, usual commercial-grade dishwashing liquid contains more trace impurities such as peroxide salts and carbonyl compounds from manufacture compared to the laboratory-grade sodium dodecyl sulfate. Third, dishwashing liquids, a type of non-ionic detergent, also tend to oxidize upon exposure to air or UV light, which results in their loss of potency as solubilizing agents. Fourth, the use of a centrifuge at 6,000 rpm also contributes to the pure DNA isolate from the bovine spleen. After DNA is precipitated with the addition of ethanol in the presence of salt, centrifugation separates cell debris such as proteins, lipids, and cell membranes from the mixture, leaving DNA pelleted at the bottom, which lessens the possibility of contaminants being present in the isolate. The bovine procedure also had the washing step with ethanol for the DNA isolate, which removes remnants of salts which may still be present, while the cheek cell procedure did not have this. It should be noted though that another likely contaminant in DNA isolates is RNA. Both methods made no mention of removal of RNA, commonly done with the introduction of RNase in the solution. Thus, the isolates from both cheek cells and bovine spleen may still contain RNA.
There are several qualitative tests available for ascertaining the presence of DNA. Particularly, we test for the components of DNA, namely the deoxyribose, purines, pyrimidines, and phosphates. We propose 5 color tests for these components. Shift test for deoxyribose, Disha's test also for deoxyribose, Wheeler-Johnson's test for pyrimidine, the silver test for purines, and the molybdate test for phosphates. Here is how these tests can be performed in order. Before any of that though, acid hydrolysis of the polynucleotide must first happen for us to break the DNA into its components. With an unhydrolyzed and hydrolyzed sample prepared, We can now perform qualitative tests, starting with Schiff's test for reducing sugar, that is, deoxyribose. If there is some magenta color change, we may move on to the next. Then Dishay's test for deoxyribose, whose positive result means a blue color change. Then the test for purines that gives white precipitate. Then Wheeler-Johnson's test for pyrimidine that gives purple precipitate. Then the molybdate test for phosphates that gives yellow precipitate. If negative results were to be found, that is, no color change or precipitate of interest was observed, then we must stop and go back preparing DNA samples. Perhaps the sample was damaged during hydrolysis, or there were mistakes in the procedure. Let us discuss the procedures for these steps. Acid hydrolysis involves adding a strong acid which will completely hydrolyze DNA into deoxyribose, purines, pyrimidines, and phosphates. We may do this by first pouring DNA sample in a test tube, then adding sulfuric acid. Subject it to boiling and adding base, say ammonium hydroxide, to neutralize the sample. Then we may proceed with the Schiff's test. The Schiff's reagent is shown on the figure on the right. Deoxyribose, which is commonly known in its ring form, spontaneously converts into the open chain form, which has an aldehyde group. This aldehyde group can reduce the Schiff's reagent, forming a magenta-colored complex called the Schiff's adduct. For this test, place amounts of hydrolyzed and unhydrolyzed sample, as well as positive control of 1% DNA, and negative control of distilled water in 2 separate test tubes. Add 10 drops of the reagent and observe 4 magenta colors in the hydrolyzed, unhydrolyzed, and positive control samples. The Schiff's test yields positive also for ribose or any sugar that can have an aldehyde group, so we have merely ascertained the presence of a sugar. To tell if the sugar is indeed deoxyribose, we do a deoxyribose-specific test, known as the Dishay's test. This involves reacting deoxyribose with a strong acid to convert it into furfural, which can react with diphenylamine to form a blue-colored complex. To do this, we prepare the hydrolyzed and unhydrolyzed sample, as well as the positive and negative controls in a test tube. Then add 1 ml of Dishay's reagent, that is diphenylamine and strong acid. Place the test tubes in boiling water, and then observe blue color changes in all except the negative control. This may take 5 to 10 minutes.
Next is the silver test for purines. This involved the formation of a purine-silver complex which shows up as a white precipitate in the test tube. To the hydrolyzed and unhydrolyzed samples and to the controls, we add 10 drops of concentrated ammonium hydroxide. This is for mediating the base-catalyzed hydrolysis of the glycosidic bonds between the purines and deoxyribose. For the hydrolyzed sample though, this is just extra measure. After that, 1.0 molar of silver nitrate is added, and expect white precipitate for all except the negative control. The Wheeler-Johnson test is specific only to cytosine and uracil. In this experiment, the target is cytosine. This involves reacting cytosine with bromine in aqueous solution to form dibromohydroxyuracil. This is followed by a reaction with excess barium hydroxide to form dialuric acid or another form known as isodialuric acid. This product appears as a purple precipitate. Again, we prepare in test tubes the hydrolyzed sample and the controls. Add 10 drops of bromine solution, then an excess of barium hydroxide. After that, wait for purple precipitate in all but the negative control. The molybdate test for phosphate involves incineration of the samples, which releases the phosphates from the DNA. The residue dissolved in water is reacted with ammonium molybdate in the presence of nitric acid, which provides apt conditions for the reaction. The product is this very big molecule shown on the right, called ammonium phosphate molybdate. First, we incinerate hydrolyzed and unhydrolyzed samples and controls in separate crucibles, followed by addition of water and then filtration. The filtrate is mixed with ammonium molybdate and nitric acid. Expect yellow precipitate in all except the negative control. If all of these qualitative tests yield positive results, then The presence of DNA from the isolation is certain.
In conclusion, DNA was successfully isolated from cheek cells using household materials. The isolated DNA samples were observed to be small in amount and to have fibrous, translucent white appearance upon precipitation in ethanol or isopropyl alcohol. The mode of actions of the reagents for the DNA isolation from bovine spleen was compared with those of DNA isolation from cheek cells. Possible qualitative tests to determine the presence of reducing sugars, deoxyribose, purines, and phosphates have also been described. It is then recommended that room temperature isopropyl alcohol be used for future DNA isolation from cheek cells using household products to avoid accumulation of salt precipitates. Qualitative analysis of the DNA isolates is suggested to ascertain that DNA is indeed present from the isolated samples. Also, tests for determining the purity of DNA isolates for the cheek cell or bovine spleen isolation may be done. Again, we are Group 5 and this is isolation of DNA from cheek cells. Thank you for listening and we are now ready for your questions.